Hello student, today we are going about the point that is the classification of bacteria. Before we have seen the kingdom Monera, that kingdom Monera having all the bacteria. Now in bacteria there is a evolution takes place. That time on the basis of evolution we can classify this bacteria into the two groups. That first group, that is Archaebacteria, this one Archaebacteria. Second one is the Eubacteria, Archaebacteria and Eubacteria. In that, first of all, the Archaebacteria. Archaebacteria means what? Archaebacteria, archaea means ancient time. In ancient time there is a evolution of bacteria. There is a first time there is a formation of bacteria. At that time, that temperature, about that time temperature means condition of our earth, that is very extreme condition, extreme temperature there is. So such a extreme conditions there is the origin of bacteria, and that bacteria we called the Archaebacteria. Now these bacteria are differentiated from the other bacteria on the basis of their cellular structure. About the cellular structure of this Archaebacteria, the cell is very simple, but their cell structure very complex one. About the cell wall, suppose we consider the cell wall of this Archaebacteria. That cell wall, it is made up of some protein and the pseudopeptidoglycan compound. Protein and pseudopeptidoglycan. This is the composition of cell wall of this bacteria. Which bacteria? Archaebacteria. Then that also having the DNA. There is also the reproduction method. There is also the mode of nutrition. This is the first point. After that, this bacteria found in extreme environment. Extreme environment, those environment there is high conditions like high temperature, high salt, high acid. That condition we called the extreme environment. Extreme environment means what? The extreme environment means the condition having the high amount of or level of that content, like high temperature, that is extreme temperature we call. So this bacteria called the extremophiles. Extremophiles. Extremo means extreme environment, extreme condition, that is called the extremo. And philes means fields, fields, that means the loving, that love that bacteria to that conditions. So that is called the extremophiles. Now these extremophiles bacteria, they are found in a variety of places, means not as one place, variety of places like volcanic area, also in the salt lake, and also in the hot springs. Their locations where these extremophiles are found. Now these extremophiles means this Archaebacteria have the ability to withstand, means to stable at any environment. At any environment they can survive. So that is called the extremophiles, means conditions whether it may be the increasing, there is no problem of such a bacteria. So these bacteria are classified into the three groups: halophiles, thermophiles and acidophiles. Halo means salt. This halo means what? Salt. And these philes means love, salt loving condition. Salt loving condition means in the salt such a bacteria are found. That bacteria group belongs to these halophiles. Bacteria can withstand high salinities, that is called the halophiles. So first group is the salt loving. Second, thermophiles. Thermophiles means what? Thermo means temperature. Thermo means temperature. And philes means love. Temperature loving bacteria means at high temperature such a bacteria can live. Then third, acidophiles. Acidophiles means acid loving, means this bacteria can present at high level of acid. Means such a groups are due to their ability to withstand. Withstand means they can stable at high salt concentration, at high temperature, at high acidity level, they can stable. So such a groups are produced in the archaea bacteria. Out of these, here is given the example. Suppose the methanogenic bacteria, that is the thermophile bacteria. One example is given, that is the methanogenic bacteria. Now this bacteria present in the intestine of the ruminant animals. Ruminant animals like a cow, buffaloes, in that animals, in their stomach, the methanogenic bacteria present. Now this bacteria helps for the production of methane. Methane, that gas is produced by this methanogenic bacteria. One more example is the thermophile, that is a Thermus aquaticus. This bacteria is also important, means this bacteria can withstand at 100 degrees Celsius temperature. So this is the one of the group of the bacteria. Now second one is the eubacteria. Second one group is the eubacteria. Now eubacteria, that bacteria it is we commonly call the true bacteria. Eubacteria, that we commonly called the true bacteria. Means on the earth, first bacteria which is present, that one is the archaea bacteria. In our earth, first organism, that is a archaea bacteria, and that is autotrophic. That bacteria showing their mode of nutrition autotrophic, then their bacteria evolve into the eubacteria. Now eubacteria we commonly called the what? The true bacteria, right? And second, the organization of these bacteria, eubacteria, their cell wall is made up of peptidoglycan. Cell wall that is made up of the compound that we call the peptidoglycan. Now about their location, about their habitat, they are found in everywhere in the nature. Everywhere they can found. Means they found in air, soil, water, everywhere they are found. But they cannot live at extreme conditions. They cannot live at extreme conditions like a archaea bacteria. Now they are found in soil, air, water, even on our skin surface. This eubacteria also found. Now their mode of nutrition, that is autotrophic or heterotrophic. Means some bacteria can prepare their own food, so such a bacteria is called the photoautotrophic or chemoautotrophs, and some bacteria are showing the heterotrophic, means it depends on other for their nutrition. Heterotrophic, that is the mode of nutrition. Mode of nutrition means how they obtain their food, that we call the mode of nutrition. Okay. Now this autotrophic bacteria may be classified into the two groups, photoautotroph. Photoautotroph means with the help of sunlight they can prepare their own food, and this another group is the chemoautotrophs, means by using the inorganic compound they can prepare their own food, that we call the chemoautotrophic bacteria. Chemoautotrophic bacteria. Now photoautotrophic bacteria, their example is the one of the sulfur bacteria, green sulfur bacteria, that is the green sulfur bacteria. That green sulfur bacteria can prepare their own food with the help of sunlight, and chemoautotrophic bacteria, that we call the sulfur bacteria or chromatin, that bacteria can use the inorganic compound to prepare their own food. Sulfur bacteria. This is the mode of nutrition. And about the heterotrophic bacteria, there are again classification, that is, they are decomposer, they are the decomposer, saprophytic, means they can decaying the dead material, that bacteria can decaying the dead material. Decomposer, saprophytic, that is the heterotrophic mode of nutrition of the bacteria. Then these bacteria are prokaryotic. All these are the prokaryotic. First things, these are the prokaryotic. Then their genetic material is a very typical prokaryotic. Genetic material about this genetic material is typical prokaryotic. Typical prokaryotic means that is the nucleoid present, means DNA is present, they are freely suspended in the cytoplasm. There is no well organized nucleus is present. Now these photoheterotrophic bacteria, they having the photosynthetic pigments like chlorophyll A, B, carotene, xanthophytes, that photosynthetic pigment present in this photoheterotrophic bacteria. Now this about photoheterotrophic bacteria, there is the heterosis cell present for the nitrogen fixation. About this one, photoheterotrophic bacteria, there is a presence of the heterosis cell, and that cell help for the nitrogen fixation. Why? Because they have the thick cell wall. In the heterosis cell, there is the presence of nitrogenase enzyme. About this point one, heterotrophic bacteria, I am saying to you the heterotrophic bacteria. About heterotrophic bacteria, these bacteria are the multicellular filamentous form. They present in the fresh water, first thing. Then they having the photosynthetic pigments. Then they having the heterosis cell for the nitrogen fixation. About heterotrophic bacteria, but about the heterotrophs, they are mostly present. Means compared to these heterotrophic or heterotrophic, these are the abundant. These are abundant, means their number are more than these bacteria compared to the heterotrophic. This is about the eubacteria. Now their functions. About the function of these eubacteria, they are very useful because they act as decomposers. Decomposers means what? They can break down complex substance into the simple substance. So they act as a decomposers. This eubacteria acts as a decomposers. Decomposers means they can break down the complex molecule in a simple form. So such a bacteria are very important to form the minerals. मिनरल फॉर्मेशन के लिए ये जो बैक्टीरिया है ये बहुत इम्पोर्टेंट है. Then some of the bacteria are anaerobic bacteria. And these anaerobic bacteria can help for the production of some product. Now here is the example given. Anaerobic bacteria example. Anaerobic bacteria example, that is a lactobacillus. Lactobacillus. Now this bacteria is very important. Why? To convert milk into the curd. Milk to curd. Milk converted into curd with the help of this bacteria. Which one? Lactobacillus. And that curd is the product is formed, and that is very important product for the human being purpose. This is the second function. Third, some bacteria are useful for the nitrogen fixation. That is Azotobacter. Now azotobacter is the bacteria that is useful to fix the atmospheric nitrogen, to fix atmospheric nitrogen. That atmospheric nitrogen is very important for the plant to the formation of amino acid. So this nitrogen is in the form of actually gaseous form, is in the form of gas. Near about 78% nitrogen present in the atmosphere in the form of gas. So such a important element is required to formation of amino acid, and that can be obtained by this azotobacter, which is present in the soil, Again important, there is the formation of antibiotics. Already we know the antibiotics means the substances which kills the other organisms, harmful pathogens, that we call the antibiotics. Now such antibiotics are obtained by the bacteria. Streptomyces is the bacteria. Now this bacteria is used to form the antibiotic production. So this is the functions. Then one more example, Pseudomonas, that is helpful for the degrading the oil and composting the oil. All these are the functions. Pseudomonas, this is useful to composting and degrading oil. They can break the oil molecules, they can compose the oil molecule. These are the useful bacteria. About the useful bacteria की ये function है, acts as decomposers, anaerobic bacteria, azotobacter, streptomyces, Pseudomonas. So many bacteria, they are useful, but also other side is there is also the some bacteria are pathogenic bacteria. They are harmful to the human beings, they are harmful to the animals, they are harmful to the plants. So other side of the bacteria, there is a pathogenic bacteria, we call the pathogenic bacteria. Now this pathogenic bacteria, pathogenic means disease causing. Now this disease causing bacteria is also present. Now that bacteria are some of the examples of the pathogenic bacteria. Fifth point, pathogenic bacteria means that causes the diseases. Here one of the disease we know, the typhoid. Typhoid is caused by the one of the bacteria, and that bacteria is called the Salmonella typhi. And this bacteria is present in the contaminated food and water. While we are eating any contaminated food and water, that time there may be the causing such a typhoid disease. Then next one that we commonly know, the cholera. Cholera is also the bacterial disease, and that disease is caused by the bacteria that is Vibrio cholerae. Vibrio cholerae, it is the bacterial name. This bacteria causes the disease. The next one, tuberculosis. Tuberculosis disease is also caused by the bacteria. That bacteria is called the Mycobacterium tuberculosis. This is the causative agent. Separate underline, why? Because that is the scientific name of that bacteria. Then tetanus, this is also tetanus disease, that disease caused by the bacteria. Now that bacteria is called the Clostridium tetani. This bacteria is present in the soil in the form of spores, tetani. These all are the pathogenic bacteria which causes the disease. Means some of the bacteria are useful, some are harmful. Okay. Then one more bacteria is given, that is very important bacteria. We know that mycoplasma. Now this mycoplasma is one of the bacteria actually, and this mycoplasma is a exceptional case of the bacteria. Already we know that all the bacteria having the cell wall, but this mycoplasma have not cell wall. Mycoplasma, this is one of the bacteria, they have not cell wall, means they are without cell wall, without cell wall. Now this mycoplasma is in the that is given, there is a term, that term is called the PPLO. This is the particularly term used only to the mycoplasma. Why? Here P means pluro, P means pluro, then again another P, that is a pneumonia, pneumonia, and L means like, L means like organism, O for organism. This is the pleuro pneumonia like organism, pleuropneumonia like organism. Now this bacteria, it is the smallest of living forms on our earth, smallest living form, remember this word, smallest living form. In your exam they may ask though, what is the smallest living form on the earth? That time this is the mycoplasma. But they may ask though, what is the smallest organism on the earth? That is bacteria. Organism, if they ask organism, that time remember the bacteria. If they asking the living forms, which is the smallest, that time this one is the mycoplasma. Now this mycoplasma can survive without oxygen. Survive without oxygen. Without oxygen they can survive. They can resist to the antibiotics. They can resist to the antibiotics. Why? Because cell wall is absent. Due to they can resist the antibiotic. This is all about the, on the basis of evolution, bacteria can be classified into two groups, Archaebacteria and Eubacteria, both having the some silent features, characteristics. And finally last point we have seen the mycoplasma. Okay, in the next part we will see about the kingdom Protista. This is the kingdom Monera. Okay, thank you.